Okay, can you hear me now, guys, online?
Sorry about that.
Perfect. Sorry. So, as I was saying, you can simulate like molecular dynamics people like me did for generations, protein by protein. But at a certain point, you need to have a mean to put this information together to understand how the overall process work. And this is what I mean going to the subcellular model. Because, I mean, staying molecule by molecule, it's interesting. You got a lot of information about the properties of each single player of okay, what you are looking there is a neuronal cascade of a cascade, but you don't get the overall idea of what is happening globally to the cascade and what is the final outcome. So, if you manage to do this, you can really go toward precision therapeutics, diagnostic, and precision medicine. Why? Because mutations that are what render each other different from others happens at the level of the gene and are expressed at the level of the proteins, most of them. So you need to model this mutation at molecular level. But then how this mutation impact the overall cascade is something that you need to understand. And if you understand this, then you can really design therapeutics and in particular drugs that are tailored for that particular mutation that the patient has. And so this is the click here. So we started with the first member of this I mean what usually is the first player of a neuronal cascade. That is a receptor with a neurotransmitter. In our case, the receptor is OG protein couple receptor. Now, usually you have, as I said, a neuroactive ligand binding to a receptor proteins that initiate a cascade that give an impulse to the brain. Now, for a lot of years, people like me, like in silico molecular modeling, only had one readout. So, does the neuroactive ligand bind the receptor? Okay, yes, this you can do very well with the technique. We can predict if a ligand binds or does not bind the receptor. We can predict the potency. We can predict the kinetics. We can predict almost all the thermodynamic and the kinetics properties of a ligand binding event. But does this count at uh, at subcellular level? Well, not really, because okay, your neuroactive ligand binds the receptor, but then what does it happen? So there are multiple cellular pathways that can be started by the same G protein couple receptor, so by the same receptor. Which pathway does it start? Do I know just knowing the binding of the ligand? No, I don't. Okay, there are some ligands that I know that promote some cascade, but imagine that I do a drug design campaign. I run a screening. I have a set of molecules that were never tested. How do I know which cascade they start? I only know from my perspective is they bind or not. But does this tell me which kind of cascade they start? No, absolutely no. And this is what we were trying to work about. So the first things that we try to understand, I mean, GPCRs are complex receptor. I will not enter into details, but they have different level of activation. But let's treat them in a very naive way, like a molecular switch. Let's treat them like we have agonists that activate the receptor and antagonists. This is absolutely not the reality because we know that the GPCRs have tons of intermediate states, and but just to for the sake of let's say of academic purpose. So let's just try to understand what does define a GPCR agonist and an antagonist. At the end, they bind in the same place. They are usually very similar. So from a binding point of view, there is not really too much difference. So we try to understand if we can differentiate an agonist from an antagonist just looking at the way in which they interact with the protein. So what we did, we took a sample receptor. In this case, I think it's the adenosine receptor. We study all the known agonists and antagonists and the way in which they bind. We build up a machine learning model. I think there was a simple random forest. And we were able to somehow have a model able to distinguish ligand binding like known agonist and ligand binding like unknown agonist. What you can see here is a confusion matrix that tells you that simply it tells you, I mean, here there are the molecule that were recognized correctly as a antagonist, the molecule that were recognized correctly as a agonist, the molecule that was wrongly recognized as antagonist were agonists, and vice versa. The molecule that were wrongly recognized as agonists, but in de facto were antagonists. So this is, you can see, is still a very good classification. By doing so, we end up with a set of completely new molecular entity that we thought could be antagonists of the receptor. But are they really antagonists? So far, we just look at the receptor. We didn't look at the cascade. So in reality, we have this fantastic set of molecules that we want our experimentalists to try, but we don't really know. So we only know that they bind similar to an antagonist, but we don't know if they are really antagonists. We don't know if they will really activate the antagonistic pathway of the GPCRs. So how can we try to predict this? Well, system biology comes in our help because it's been one of the oldest way to schematize cellular cascades in general, not only neuronal cascade. And it usually are differential equation that takes as an input thermodynamic and kinetic parameters. So I'm talking about KD, K on, K off. All these parameters you can get, as I told you, from the simulations. You need, of course, someone that somehow put down the differential equation for you, because each pathway has its own system of differential equation, and this needs to be carefully tailored for the pathway that you want to see. But if you have, in our case, we have the ones of the adenosine receptor. This was why we chose that particular receptor. If you have it, you can try to derive all these parameters from molecular dynamic simulations, feed it to a system biology model. That means, again, as I said, a system of differential equation, and try to calculate some kind of response. So this is doable. What is the main problem here? That you have a system of differential equation depending if it is an agonist or antagonist. So also here we fall in the same problem. You need to know in advance if it is an agonist or an antagonist, because you need to choose the correct system of the equation. So if you have a completely unknown ligand, and I'm not talking ligand in the literature, but if you have completely unknown ligand, like the ones coming out from a screening procedure, from a virtual screening procedure, which system of the equation do I use? And here was the kick of our let's say work. So we run molecular dynamic simulation. So we extract a lot of structural information and dynamical information of the receptor. We perform ligand screening. We detain only the ones that were binding or that were predicted to strongly bind to the antagonist. To the inactivated form of the receptor. So supposedly they were, let's say, happy to be in an activated structure. And then we introduce for the first time this AI-based agonist-antagonist discriminator, because this tells us the probability of our unknown ligand, at least based only on the way in which it was binding to the receptor, if it was an agonist or an antagonist. So better, not if he is, but if it has a high probability to be an agonist or an antagonist. And based on that, we can select which, let's say, biochemical network model to use. So we did so, and we end up in all the 10 ligands, only one actually was predicted to be by our biochemical network model on antagonist, and our prediction was surprisingly good. This is the prediction, the predicted binding curve of our lead candidate, and this was the ones that our experimentalists detect. So it was really a good agreement, and the fantastic things but this is from a chemical point of view. I know that not all of you are chemists, but chemists get very excited because you see this molecule has nothing in common from a chemical point of view to all the known antagonists of the adenosine receptor. So it was a completely new molecular entity that we were able to find in a completely unbiased way, just putting together molecular dynamic, artificial intelligence, and system biology. So in some way, I'm not saying that we were able to go completely at the subcellular level, but we were able to identify the main features that allow us to say something about the cellular pathway. And this for us was relevant. And of course, it went to a publication. So this is a chemical science that was published 2023, so not so much time ago, and he speak about this approach. Now, why we were so excited? Because when you do drug design, in silico drug design, as I said, you usually always look at the single target of your drug, and this is cool because you can deal with genetic mutation. You can be highly specific, but you do not understand what the drug is doing in the context of the cell. If you go for the system biology, you can have this understanding, but then you lose all the molecular details, so you cannot really model a mutation, or you cannot really be precise in your drug. So bringing together this is what allow you really to go toward precision diagnostic. Now, this tool is available in the EBRAINS set of tools, and thanks mostly to the work of these two researchers, Alejandro and Rui, that made, let's say, a user-friendly toolkit for using this system biology. We call it structural system biology because it's based on the molecule and is available in the pool of tools that are on the EBRAINS. Now, we were very confident and very happy about this result, and we thought, can we go from molecule to understand something about plasticity? So can we do something about can we do something that we call molecular plasticity? This is a little bit unusual term. Okay, the idea came because we were in a fantastic network that was dealing with pain transmission and neuronal plasticity. The title of the consortium was Sodium Channel Related Pain Disorder from Molecular Mechanism Toward Personalized Treatment. And if you look a little bit at the people involved there are people like me that perform a simulation, but there are also medical doctors and electrophysiologists, psychologists. So it is really a very, I don't know, variegate ensemble of people with completely orthogonal expertise. So now, why we are talking about the neuronal plasticity when we talk about pain? So pain transmission usually happens to this nociceptor neurons that detects the harmless the pain stimuli, and the signal then travel from to the spinal cord and then ascend to the brain. So this is how the pain is perceived. So pain is one of the very most common example of neuronal plasticity because the system is meant to be protective, but to be protective needs to be adaptive because if you touch something spiky and you get and you get hurt, you need to learn not to touch it. So I mean, it's by definition a process of plasticity. So neuronal plasticity means, in this case, that pathways need to adapt based on activity, injury, or experience. So pain circuits can strengthen, weaken, reorganize. And so the pain perception is something that is dynamic and not static. That's why we are talking about plasticity also when we talk about pain. So it's one of the process, straightforward process of learning. So now, what can modify this perception of pain at molecular level? In the same days, there was an interesting paper coming out that the title was Membrane Cholesterol Depletion is a Trigger for Sodium Channel Mediated Inflammatory Pain. So the author discovered that when there is, I mean, NAV 1.9 is mostly on the epithelium, and so they were doing experiment on the pain perception on the skin, and they discovered that when there is an inflammation in process, so when they were doing the experiment with mice, but when there is pain involved and there is an inflammation at the level of the skin, if they go looking at molecular level, what they see is that the channel becomes somehow hyperpolarized, okay? And it become hyperpolarized. They suggest, they didn't prove it, but they suggest that it become hyperpolarized because it is kicked out from the so-called lipid raft. Now, the plasma membrane where the receptors sit is not an homogeneous medium. So there are regions that are higher in cholesterol and regions that are lower in cholesterol. The regions that are higher in cholesterol are called lipid raft, and usually is where most of the channel cluster, because they are considered some kind of hotspot region. And they discovered that in the presence of an inflammation, the channel is most probably kicked out from this region, and this generate hyperpolarizability. So we thought, okay, interesting. They didn't. It's an interesting theory. They didn't test it. They didn't. So this was their opinion. What about we don't do the same? So we of course focus on not on now 1.9 where we were looking at chronic pain. So we took as a example another sodium channel, namely 1.7, that is actually considered the gatekeeper of pain. It is involved in the endings of all pain-sensing neurons in the nociception system and play really a critical role in the generation and the conduction of action potential. So we focus on NAV. And we started to do something very simple and very basic for a molecular person. So we started to simulate this in different cell environment. Because I don't know if you're aware, but your plasma membrane change a lot. Change if you are in a disease condition. For instance, it changes a lot with cancer, with neurodegeneration. But even if you are a healthy person, it changes completely with age. So the plasma membrane protein of little kids is completely different than the ones of an adult. So the composition changes. So this is again another process that can regulate plasticity because, also aging is play a role in plasticity. And if the membrane protein changes, then you can have a role. So we simply simulated this fantastic ion channel in different membrane environment. This is just a scheme to show you how the channel is done. So you see there are 4 groups of helices that fold in this kind of tetrameric structure. This is an ion channel, so ion should pass, and ion pass through this hole in the middle. But is also, again, a voltage gate, so it means that it needs to sense the voltage of the membrane, and these other 4 elements here are the ones that are responsible to sense the voltage. So what we did, we just try to answer to three questions. So does membrane composition affect the dynamics of the channel? Are these effects sufficient to impact on the channel's activation and inactivation? And can this affect also the channel drugability? Because I mean, if I have an hyperpolarizer channel, the drugability will, I mean, the ability to bind the drug will be different. So we did this, and we find at molecular level in our simulation that cholesterol indeed localize around all the hinges point of the channel. What does it mean? If you block the hinge point of a movement, what does it happen to the channel? The channel is more rigid. It cannot really activate or inactivate rigidly. So when you remove the cholesterol, the channel is much more flexible because it has the hinge points free. It's like if I put something here in my elbow, I cannot close it anymore so easily. But if I remove, my elbow is free. And the same happened with cholesterol. Cholesterol, that is this red balls that you see there, somehow cluster around the main region. And so when there is cholesterol, the channel is very symmetric. But when there is not cholesterol, the channel lose the symmetry. So the hole in the middle is not square-like anymore, but it become more rhomboid. And this is actually being connected with the activation. So what does it mean? That without cholesterol, the channel becomes somehow more flexible, more loose. In other words, hyperpolarized. Is this true? So our fantastic experimental collaborator did this experiment. I mean, it's very easy to test the impact of cholesterol because, you just put a chelator in the cell and that progressively remove cholesterol so you can see which is the effect on the channel conductance. So what we did, what they did is exactly this. And what they discovered is that cholesterol depletion, so when you don't have cholesterol, you increase current density, you hyperpolarize the voltage dependence activation, and you even change the kinetics of the channel. So in other words, the channel become hyperpolarized. So our colleagues of the paper that I showed you at the beginning were right. So channels become hyperpolarized when they are in a low cholesterol environment. And this can happen because you have an inflammation, because you are aging. So hyperpolarization could be a general mechanism by which, so in particular low cholesterol, modulation of cholesterol could be a general mechanism by which the gating of a channel is affected. So, and this change, of course, now this is just a picture about the volume of the most important binding site. So it changed also the drugability. Several papers came out from these studies. I just reported the two. One is about what I just told you, the depletion of membrane cholesterol modifies structure, dynamic, and activation. And we propose that changes in membrane properties might be a general mechanism by which cholesterol depletion alters the gating of the sodium channel. But we also discovered that it could be a way to amplify genetic mutation. And this was a paper that we published together with a medical doctor, where we showed that some mutation found in kids of this particular channel can be amplified in the absence of cholesterol. Now, what is about neuroplasticity? So, you see, at the end, in the normal state, the cholesterol as a given, so in the normal membrane with let's say the correct content of cholesterol, you have or polarization of the channel that is healthy. But if you have any kind of inflammation or pain process, essentially what happens is that the channel become hyperpolarized, and so they increase the firing, and you might have a neuron excitability. So in this way, it was for us a way to prove that understanding of phenomena, or set of phenomena at molecular level can tell you something about how, let's say, plasticity work in case of pain transmission. Now, these two examples, as I said, for us were extra extra interesting and brought us a lot of publications. But still, they were not the real thing. So in the sense, we find connection at higher scale, in one case at the subcellular level, in this case really at the neuronal level. But the connection were established between, let's say, simulation and experiments. And we wanted to understand if we can instead connect really two different simulation schemes. And this brings me to the last part of my seminar, that is a work that we did together with Sandra here. And it's from atom to neuronal spike. So this is a paper that was published recently. And you see there are people from ULISH, and you know Sandra. And let's say that Sandra and Paolo were the main minds behind, but Thorsten and Anna and Naumann and Vincenzo were the ones that really practically did the job. And never forget, because one thing is organizing a project and having the idea, but one other thing is then the people that realizes you need people that realize your idea, especially especially extra good scientists like in this case. So I told you that if you look at, people that knows me and that knows the work that I do and the works that Paolo Carloni does, they know us for molecular simulation people. So we simulate things in a box, okay? But if you think about Sand and Thorsten, then they are usually associated with Arbor, so they are usually doing neuronal simulation. So how do we put these two things together? So this was the last work that we tried to do. So traditionally, from molecular simulation, what do you have? Atomic detail, but you don't have any neuronal output. And from neuron models, you have spikes, but you have very, very simplified channel behaviors. So we try to bridge in this paper these two simulation framework, and we manage, and I will show you how, of course, a theoretical point of view. I will not enter in the details of the equations. So what do you get, or what you can get from molecular simulation, from what we call the molecular layer? So you get, if you simulate, for instance, an ion channel, you can have ion permeation and conductance. And if you coarse-grain this, and if you, top up a little bit the scale and you make a coarse-grain model with Monte Carlo that is still considered, in any case, a molecular layer, you can get the channel gating dynamics. And the outputs that you can get from these two schemes are usually the current, the conductance, and the state transition. Okay, this is what you can get at the molecular level. So what you get from Arbor, and Sandra, please correct me if I say something stupid. So it's a morphological detail than neurons. In this case, for instance, we use pyramidal cell that are simulated with outputs and with Arbor, and the outputs are membrane potential and spike traits. Okay, this is what you get. So we try to link these two levels in two ways. The first things that we try to do, that we call case one, is a simple, let's say, I would say, bottom-up approach. So we simulate an ion channel, in this case was the AMPA receptor, and we measure the change in conductance of the wild-type ion channel with respect some specific disease-associated variant. So this is so we get this kind of conductance, current and conductance, and this were fed to inform morphological details and model of pyramidal neurons simulated through our board. This determined the neuronal spike. With Arbor, the membrane potential was calculated and the spike. So what we understood is that when you have disease-associated AMPA variants, you can impact the neuronal excitability. Now, the second example was a little bit more tricky because it really tried to create a feedback loop. In the second example, what we did was Monte Carlo gating. So we have a very simplified representation of sodium and potassium channel. You see these are these squares here. Around there are little squares, different types of red that sense the surrounding voltage. So they are light red if they are resting, or they are bright red if they are, let's say, activated, and they even sense somehow. You see, there is another composition. They even sense the lipid composition because, as you have seen in my previous talk, it is relevant for membrane protein. So what did we do here? A little bit different approach. So the Cosgrave Monte Carlo gating predict the current. Arbor calculates the resulting membrane potential and the spike generation, but the voltage then was really feedback to the Monte Carlo simulation of the voltage gated channel. So in the second example, we really have a sort of bidirectional coupling of current and membrane potential. So the key novelty is that so from one side you have this dual coupling between the voltage and the channel state interaction, and also that the other model novelty is that you can include the lipid membrane. So just to explain a little bit better, so channel states can be open and closed. You can generate ionic and calculate ionic current. This was given to Arbor that calculate the membrane voltage and the voltage dependent gating rates, and this was feedback. So change again, the channel state change again, and then you can restart. But this is at the Monte Carlo level, and this is instead, for instance, at the Arbor level. So in other words, while in the first case you are mostly feed forward, the second case try to introduce some kind of dynamical loop. So the neural voltage control the ion channel control the voltage. And this is exactly how it is supposed to be. So in other words, what was introduced here, and again, I hope to be clear, is that instead of having Arbor following, let's say, a deterministic equation, you get to follow the output of Monte Carlo, that is where channels randomly open and close based on physics. So was the main, in simple term, the main let's say breakthrough that we did with this paper, and it is extra important because for the first time we had an input that was not deterministic but was based on physics, Monte Carlo based physics. So clearly a lot of limitation, but still it was the first time. So we have these two cases. And in principle, now we can ask how does a mutation change the gating at atomic level? How does it affect spike timing? And how does membrane composition affect excitability? So in principle, we can now answer to all these questions. In practice, we are still working on that because, parameter transfer between the scales was not a trivial operation to do. It took a lot of time and is still very much system dependent. So we are not so smooth yet in this in these things. And as I said, we are still limited to specific channel system because, of course, it's not that we have all the models ready for all the channels in the world. We have only few channels that have been modeled and that work smoothly. But, at the end, you want to show that it's possible, at least in one case, because if you show that it's possible in one case, there is hope to then extend this to a more general simulation framework. So I hope to have convinced you that at the end of all this year of working, we probably did only the first line, so molecular change and alter the neuron firing, and we started also to try to understand how molecular shape can somehow have an impact on plasticity with the work on cholesterol. But there is still a lot to do. But we still think that it is, to answer my question here, is it worth the effort? Yes, we think that this is worth the effort because this really allowed to go to push medicine and drug design in direction that were unthinkable before. So with this, I would like to acknowledge, so here there are the experimental group that, Sandra, Thorsten, Paul, and the other people I already acknowledged during the seminar. These are the students that did the computational work and the postdoc that did the computational work, Simone, Alessia, and Jonas, the funding that supported us, and thank you.
Thank you very much. Do you have questions?
Yeah, thank you for the talk. And a very specific and a very general question. The specific question is, what was the input of the model trained to see whether this might be binding as an antagonist and agonist? Is it just like the sequence of amino acids, or were the structural information?
No, what we use or protein ligand interaction fingerprints. I don't know if you heard about. So there is a way to schematize chemical interaction as a vector 0, 1, 0, 1. In other words, you can, chemistry developed approaches to translate a set of chemical interactions in vectors of 0, 1, 0, 1, 0, 1, because this can be easily understood by a computer. So at the end, what you have is like a barcode that univocally identify your molecule, and now the molecule interact with your receptor. And these are called protein ligand interaction fingerprints. So they are barcode. You feed this barcode, and I think that I can show you a very quick example of what we mean with this one. You see, this is not an interaction fingerprint. This is just a way to schematize a chemical structure, but the principle is the same. So, does the structure have benzene? Yes. So I put a 1. Does the structure has this particular chemical group? No. So I put a 0. At the end, you have something like this. And this you feed, this is something that whatever machine learning algorithm understand fantastically, because it's easy. It's 0, 1, 0, 1, 0, 1. There are tons of ways to generate this barcode. So you need to find the correct one, and this is time consuming, but you can do this.
Discovery with AI, I think it's also like a buzzword currently a bit that companies and startups try to do. And how do you see this approach more as a basis for target identification, and then would be nice to have the multi-scale level approach later on, or could it replace this? And, yeah.
I don't know, this is a difficult question because machine learning and artificial intelligence in general is as good as your data are good and the amount of data. And it is proportional to the amount of data. So AI is not a new thing. It is known since the 50s, but before there were so little data that it was never working properly. So now we have tons of data and it starts to get us results. But it's not so easy like it looks like. First of all, you can end up in a lot of artifacts, because if you don't have a cleaning database, it's really difficult. Then it's another problem is that when you have data coming from different sources, to integrate them is not so easy. So to do this for target identification, you need to put together structural target, cell biology data, toxicology data, and so far and so on. So I know that this has been done in some specific case, but I don't think that we are still at the level of generalization that like we push a button and we have the target identified. So again, it's system dependent because it depends on the data. So, and for drug design, yes, there is a lot of use of machine learning. And yeah, it's extra useful. But again, I think that you need to do this with a little bit of, I don't know, cognition of what you are doing, because otherwise you might end up in having very wrong answers. And so again, you should always go back to your data. But yes, if you have good data, if you have well-prepared data, if you have enough data, machine learning can be a big, big, big help. Absolutely.
So good morning, everyone. My name is Luhan. Thank you for the kind opportunity for me to introduce what I'm doing in Fortune Centrum here. So I'm currently a postdoc fellow in Simulation Data Lab Neuroscience led by Sandra. So we actually do computational neuroscience to understand the brain dynamics, neuroplasticity in health and in disease. So here I'm going to introduce you a project that I have been working on before I joined Fortune Centrum and after I joined, which is decoding neuromodulation through multi-scale neuromodeling. So there, I firstly used the NEST simulator. Have you ever heard about it? Okay, cool. Point neural simulator with the possibility to really run large neural network with plasticity. I moved to Arbor. I'm now in the Arbor developers team, and I'm going to showcase how we use these both tools to model TDCS, transcranial direct current simulation. So this is who we are. Our team combines supercomputing and human brain science, human brain project. We do both development and science. We support scientists in the field, and we also co-design with them. I'm currently in Sandra's lab. So in our lab, we have learning to learn methods, algorithm development, and we also study structural plasticity. This is something we're going to talk about more in detail in my following presentation. And we also have the virtual brain platform, which is a platform to model the whole brain. And we are part of EBRAINS Skills. We are developers for Arbor, and in the end, we are trying to combine different skills, different simulators to do multi-scale co-simulation. So these are the three major simulators that I am going to address more in detail in my talk today. TBB we call it at a macroscopic level, modeling whole brain dynamics, or in the mesoscopic level, like we zoom in to look at a local neural network. And the last one is Arbor. We can even have more details there. We focus on single neurons, even with detailed morphology and biophysical properties. So these are the projects that we have been working on in the lab at the moment. So getting back to the topic, neuromodulation, or I have to say non-invasive brain stimulation, is currently a promising alternative treatment to neurological and neuropsychiatric disorders. For the past 2 to 3 or 4 decades, they have demonstrated new hope to provide effective performance in treating depressive disorder, Alzheimer's disease, or help the rehabilitation post-stroke, or even enhance working memory in elderly folks. So the major tool types included transcranial magnetic stimulation. One is using the magnetic coil to trigger eddy current in the brain tissue to stimulate the brain, and that is a suprathreshold. When we call it a suprathreshold, it means it's very strong. It can directly trigger a silent neuron into firing state, like triggering an action potential there. Another one would be transcranial electric stimulation. Depending on the current you use there, it can be classified as direct current, DDCS, alternating current, TSCS, or simply use a noise format, TNS, to do the stimulation. So TES, especially TDCS, is considered as sub-threshold, meaning that if you directly deliver it to a silent neuron, it will not trigger action potential. But if the neuron itself is already active, meaning in our brain, now when you are listening to my presentation, your neuron are firing, you have a background activity in your brain. So now if this sub-threshold current delivered to this organization, it is going to trigger more activity there. So use brain stimulation as an example. If we want to use modeling to understand it, you may first ask why, because we want to better improve the performance of the brain stimulation after we fully understand it, how is the working or action mechanism of it, and how the single stimulation parameter components trigger midterm or short-term, midterm, long-term effects. So different ways to do it. If people focus on a single neuron performance, a single neuron activity inside the electric field, this is more microscopic, or people can further go to the network with large connections of the cortical column and even the whole brain. So different mechanism, if we really focus on different skills, and this actually makes simulation a powerful and interesting tool to give us insight about it. So here I'm going to introduce you to the first appetizer, my previous work, how I modeled TDCS using a mesoscopic level model. That is a spiking neural network with homeostatic structural plasticity rule. So before my work, people, when they model TDCS, they were mainly modeling membrane potential deviation inside the electric field, which is at microscopic level here. But this is complex and computationally expensive. So if we wanted to understand how this could work at inside a large spiking neural network, it was not really possible at that time. So the first simplification I did is, okay, now I don't care about the strength of electric field or the orientation of the neuron inside the electric field, because they jointly determine how strong the neuron will be stimulated. I simply focus on the magnitude of membrane potential deviation. So if the membrane potential at the soma part is depolarized or hyperpolarized, I use it to model TDCS in a single point neuron. So this gives me the possibility to now build a super large network of point neurons. And therefore, now I can model a brain tissue underneath electrode. So you see two types of montage of electrodes in TDCS. Montage meaning how you organize electrodes for the stimulation, like the conventional ones or traditional ones is two large paths on the brain, or people use a button-like electrode to really plan a local stimulation with TDCS. So the first one will be more diffuse, and the second one is really within this area around the electrode. So with this one, what kind of network I want to build there? I want something that we already know the behavior of the network. So it's like when you do biological experiments, you are not just capturing mice or rats from the wild to do it. You should know very well the genetic background of the animal. So we started with a Brunel network that has been thoroughly studied, that we know if certain parameters got changed, then what the dynamics will look like. So this way we have like a reference. When we observe any changes of the network dynamics, then we know what kind of impact our stimulation did there. So this is a network with excitatory and inhibitory neurons with an 8:1 ratio. And then I deliver TDCS, or I model TDCS to a subgroup of excitatory neurons there. And what I will look at it, I firstly enabled plasticity there in the network, because I'm not only interested in how the acute effects of the stimulation is, but also the chronic effects. Like when I stimulate the network for a relatively longer time, what will change? So now you have a plenty, like you have a long list of candidate plasticity rules there. What will change? What will you pick from them? So if you are familiar with NEST, so they have a plethora of functional plasticity rules. I hope you know the idea of functional plasticity. So this is how strong neurons got connected with each other, which is the synaptic weight. To change synaptic weights there. Another one, I'm using a different plasticity rule, is structural plasticity. So we allow neurons not only to change not changing synaptic weight, but changing the numbers of connections. So this is a rule that I use a linear rule, and we allow the creation and pruning of synapse, and it's a rather slow process. So there we can capture a longer process, and it's actually guided by homeostasis. What is homeostasis? Imagine a neuron, it tends to maintain its firing rate within a functional regime. So which is also makes sense for our brain. It's not going to be too active or too silent. And in the case of stimulation, if the activity of this single neuron goes too high, then it reacts homeostatically to reduce excitatory synaptic input by pruning the synapse, by breaking the synapse. And this way will bring down the neural activity back to the set point, the homeostatic level. Vice versa, if the neuron is firing too low, and then the neuron will be trying to grow new synapses to form with each other, compensate its activity with recurrent input. So this is the rule about. And then this rule, when it's working in a large network, it has an interesting phenomenon like that. This is a network grow to a homeostatic level. And then we stimulate a subgroup of excitatory neurons. What will happen? They will their firing rate will increase, which makes sense. And this will trigger homeostatic elimination of excitatory connections. Therefore, the firing rate will be going back to the set point. And at the end of stimulation, now the removal of the stimulation and the loss of conductivity will result in the drop of firing rate of the stimulated group again, because now there is no external stimulation and the recurrent input is also low. So this will trigger the opposite homeostatic process for the neurons to grow more connectivity, to grow synapse. So the connectivity of the subgroup goes up, and then to restore again the firing rate. So at the end of the stimulation, actually, at the end of the stimulation, after the network to relax a bit, in the end, there is a subgroup of neurons. They were stimulated and they have the high connection probability. So we call it a cell assembly. So in neuroscience, when there is a cell assembly hypothesis, is considered as the mechanism of many cognitive processes like learning and memory. Therefore, I did the same. I did TDCS modeling to a subgroup of excitatory neurons. But TDCS, there are many parameters we wanted to understand. So this is the first toy model in the field. Nobody has done that before. So the first thing will be, okay, how about current polarity? Because depending on how the electrode you put there and how the somatodendritic axis of the excited neuron inside the electric field, the actual soma membrane potential could be depolarized or hyperpolarized. So I did the same to both. One is a depolarizing current, one is a hyperpolarizing current. And interesting that it's very weak, sub-threshold, but long enough after the stimulation, we both observed a stronger connectivity within the stimulated group. So no matter it's depolarizing or hyperpolarizing current stimulation, we have a very weak cell assembly formation there. Another one is how about electrode montage? So we have to abstract a bit the situation if we wanted to study contact, because we are not really modeling the whole brain with complex geometry. So depending how large the electrode is, it may cover relatively large network within a large population of the excitatory neurons in the network, or smaller one. So this is one parameter I use there as the relative ratio of the stimulated neurons from 10% up to 70%, the systematic study. Another one is how strong it is. So the intensity of stimulation is jointly determined by stimulation intensity itself and the electrode montage. If it's more diffused, actually the electric field is weaker. If it's more focal, even the same current delivered to the brain, the stimulation might be stronger. So I did all of this and figured out that the stronger the better, the focal the better. Another one, like really interesting parameter is how do you want to repeat the stimulation? This is always like the major challenge faced by clinicians when they have to treat patients. Do they ask patients to come in the morning and in the afternoon every day for 5 days a week, or they ask them to even come for like 10 times a day? It turned out even in human study, they find that like how the interval they planned during stimulation session makes a big difference there, but they don't know why this happens. So our model for the first time provided explanation there. I repeated the stimulation, like you see 5 times there. And then we know there is homeostatic reorganization. So the firing rate was like going up and down, circularized with a repeated pattern. But for the connectivity, so every time it increased a little bit the connectivity within the stimulated neuron. Let's see, the connection probability of the cell assembly actually built up through repetition. So this is interesting. Then I tried the different interval and the relative duration between the interval and the stimulation time. So compared to a super long stimulation, the white one here, if we break it into small epochs and delivered to the brain, they actually immediately boost the connectivity. So here we assume that stronger connectivity, stronger cell assembly might be linked to stronger after effects of the stimulation. So we even did more repetitions and find that it's not like the connectivity will build up or increase at infinitum. It actually will saturate after certain runs. And then the saturation level of the connectivity is determined by the intensity itself. So we do need to have a relatively stronger stimulation to reach the final connectivity to a substantial amount. But it should better not to be too strong. Otherwise, there is a runaway unstable dynamics of the network. So this is very interesting. Polarity, intensity, repetition patterns. We have provided the qualitative explanation to human studies results which cannot be simply explained by doing human studies and animal experiments. So here, actually, if the stimulation will achieve better effects with stronger focal repetitive stimulation, this actually pointed to TMS, which is very strong, suprathreshold, and in a repeated pattern that it has pulses, and also very focal. So this leads us to have first follow-up study to increase the intensity and to model TMS using the same network architecture. That's already actually interesting that TMS is not just a stronger version of TDCS. So it's no longer the stronger the better. There is a sweet point of the intensity. So if it's too strong, then not shown here, but if the stimulation is too strong, then it's going to activate inhibitor neurons more in the network and trigger weaker effects. Another one would be we have modeled TDCS in the first model in a baseline condition, basal condition. So nothing is going on in the brain, and we just deliver TDCS, and we propose that the stronger the better, and better to have breaks in a repetitive pattern. But in real experiments, there's always something going on in the brain. And particularly one type of task is used a lot as learning, motor learning. Because TDCS was considered to boost the learning effects in formal and informal studies report. And then we wondered, okay, if people want to do that, even if it's not in healthy subjects, if they use it to, for example, to facilitate the rehabilitation or the motor learning in patients after stroke, then when to apply it. So in so many human studies, they are reported that the data, the results is highly inconsistent. Actually, if the same stimulation is delivered before, during, and after stimulation, and even long after the motor learning process, the results is highly different, even opposite. So there are some theory how to explain it, but there is never a systematic understanding of why this happens. So in this work, we further did, okay, we established a learning process in the network by triggering, again, cell assembly with external input. And then we applied TDCS there. Different parameters are interesting here. Like if the stimulation is diffusive, so like the electrode covers more areas than the target region, so we model it as a uniform TDCS, or it's a highly targeted stimulation, like we precisely target the neurons involved in motor learning or involved in the motor engram. And then we delivered this type of stimulation before learning, during learning, and after learning with different polarity, depolarizing or hyperpolarizing. And we find this is a summary plot that in some conditions that the stimulation boosts learning, and in some conditions the stimulation really hinders learning. So this is really interesting that we reconcile the state-dependent stimulation results. So when I entered the field, we established a toy model to understand, but now with time we have trying to make it to connect better with parameters used in human studies to provide an understanding of what they observed there. So this makes it simulation a fascinating tool to compensate to what has been established in the field, like animal studies and human studies. So after I joined Arbor, okay, my tool is now to advocate Arbor more. So Arbor as a simulator is different from NEST. NEST as simulating pointing neurons. And then you can run a very large neural network, like 10,000 of even 10,000 neurons is what you see in my presentation. And with different plasticity rules. And with Arbor is rather new, and you can simulate one neuron with complex morphology. We will see it later. And it is not just leaky integrated fire action mechanism. You can have really biophysical features there running by the cable equation. So we have all of these features, and Arbor is possible to use GPU, like the Jules Booster we have it here to really scale up the simulation. So now I'm going to give you an overview of the progress we have in Arbor. The first thing you have seen it in Julia's talk. So it's possible to connect molecular dynamic simulation with a single neural simulation in Arbor. Another one is Arbor is possible to host plasticity rules, like the functional plasticity rule based on the calcium concentration of the neuron. All the researchers sitting also in the audience that we learn, and we have distance-based structural plasticity enabled in our. And the last one is our as a simulator focus at a microscopic level, and we managed to connect it to TBB. If you still remember the name, it is a simulator to model the whole brain dynamics by abstracting individual areas as a nodes and represented by action function. So there we actually coupled Arbor with TBB, so we can replace one node, like one region in TBB with a network simulated in Arbor, and it allows for real-time exchange between Arbor and the TBB. The only difference is in Arbor, when we simulated, it gives us spikes, so it's a discrete events. And in TBB, activity is simulated, or the output of TBB is time series. So we have to find a way to communicate them better. With this framework, we have developed a use case of epilepsy. So we replaced one region here in the hippocampus with network simulated in Arbor. So these neurons, if we change the potassium concentration in the bath, then they can have epileptic activity like this. And then by having the co-simulation, then we saw the propagation of the seizure-like event from the local area to other brain regions. So this is a new tool and can give you the flexibility to do many things. So now we comes to one major project I'm working on. It's called Arbor for AD, Alzheimer's disease. So we really want to utilize all these features developed around Arbor to build a multi-scale model to investigate neuromodulation parameters of Alzheimer's disease. So there we wanted to really bring different levels or different scales of simulation. And the first thing will be, I want to replicate TCS in Arbor. The first thing, okay, now I have a single compartment of neuron subject to Hodgkin-Huxley dynamics, this HH neuron. If I have current injection to it, if I have a Poisson input to mimic recurrent input, how the activity of the neuron will be like. So I actually designed the four types of four phase of experiment without input as a baseline, having the current only, not really making a big difference until the current intensity is super high. Or with only Poissonian input. So there's just to mimic an active neuron with recurrent input. And the other one is, okay, on top I have TDCS. So this is replicated the subspatial stimulation dynamics there by having very weak stimulation to an active neuron. We actually managed to increase the activity of the neuron already. So Arbor is never designed for a single compartment of neuron. Then how about we really bring some complexity there? So now I'm showing you, now we wanted to simulate a neuron like this, or any neuron you obtain from your experiments or from your collaborators that they have recorded, they have, let's say, injected dye to the neuron, and then with microscope imaging, they captured and reconstructed the whole dendritic tree of the neuron and labeled as soma, dendrite, apical dendrite, and basal dendrite, and axon. And now we want to simulate something like this. That's possible. We just load the morphology file to the neuron and to Arbor to build such a neuron and apply the same Hodgkin-Huxley dynamics there. Two ways to do it. You can choose if you want to have passive dendrites or active dendrites. Active dendrites meaning even on the dendrites you have Hodgkin-Huxley dynamics there. So in this example, I'm showing you that interesting. With passive dendrites, it seems like the neuron is very silent, with the same experimental design as in a single compartment neuron, and even the Bosonian input cannot activate the neuron because all the dendrites are silent, are passive. But if we have active dendrites, the activity level really goes up now, and we replicated what we observed in the single compartment neuromodeling here. So this is something to play with morphology. And another one is that we are also not only interested in single neuron simulation. We wanted to have really large neural network. So we wanted to build, let's see, a Brunel network, and then to deliver TDCS to 10% of the excitatory neurons there. The first thing is let's try to replicate what we have in the next simulation. So here we use leaky and integrate fire neurons and delivered 100 picoampere TDCS to 10% of excitatory neurons. So this is at the baseline, excitatory neurons and inhibitor neurons on fire around 10 Hertz. And now I delivered the TDCS to 10% of the excitatory neurons. Okay, then for the stimulated neurons, their activity goes up. And then if it's gone, there's no plasticity at all. Therefore, the activity also goes down. So this is one thing. Okay, with leaf cells, we managed to replicate the network dynamics there. How about now we increase the complexity to have a single compartment of cells? And interesting, even if I have like a single compartment neuron with a radius of the soma to 9, and it looks like the network activity is no longer asynchronous irregular, like nicely as it here, but we have like a damping dynamics with a large ratio of the neurons being silent in the network. So I wonder what makes the difference. It turns out it's nonlinear dynamics of the HH neuron even at a single compartment level. So by having different Bosonian input rate, the activity can go up a bit and then drops. So there is more to consider if we wanted to have a large network with Hodgkin-Huxley dynamics, so which is more biophysical there. And I even did something just for fun. Okay, now I am adding the neuromorphology to all the individual neurons there in my 1000 neural network on my local PC. So here I have pyramidal cells with passive dendrites. So if you still remember, with passive dendrites at a single neural level, it's actually very silent. But now I put neurons with pyramidal cells with passive dendrites in the network. It goes to overexcitation. So what will you imagine if we even put passive neurons, pyramidal neurons with active dendrites? Just give me an answer. Will you expect even more crazy activity here?
You see the question again?
Yes, it's, the question is, so at a single neuron level, if it's a pyramidal neuron with passive dendrites, then it's very silent. But if the dendrites are active with Hodgkin-Huxley dynamics, it's going to be active like normal. But now I put these two types of neurons in the network, and the network with passive dendrites is already firing crazy around 250 Hertz, which is unrealistic. So my question would be, now, before I show you the results of such a large network with pyramidal cells with active dendrites, what do you think will be the dynamics there?
We don't know.
By adding more conductors, just expect to have more activity there in period.
So, okay, even more answers?
Even lower because they will separate the channel so they won't fire, so we will start having connectivity on the network and start working.
Interesting, yes. Even more guess? Of course, only two directions. Okay, I'm showing you a very beautiful raster plot here. So we have 1000 neural network with pyramidal cells what we see there with active dendrites seems to give us a reasonable regime of the neural activity. So I wondered why. Yeah. So like you mentioned, conductance or what I find would be the membrane integration, because for the same external input, I have 1500 Hz here. So having active dendrites is actually a slow membrane integration because at each segment and there is integration, and therefore it gives us delayed spikes. And therefore, like all the spikes are not arriving at the same time, so it's decorrelated or not synchronized arrival and give us a slow synchrony and balanced network. Otherwise, it is a fast integration and kind of like a positive, like closed loop and triggering further the network activity. This is also something new I learned with ABR, because before ABR, we cannot really model very large network with this complex morphology. So this is one hypothesis I provided here, and there are more to explore if you are interested. Another one, okay, now we move to an even more complex network architecture. So in NEST, always point neuron model, there is a famous model which models it's called the microcircuit models the cortical column with different layers, different cell population and connection probability. So this is the dynamics of raster plot at different layers in their model, which fits nicely to the physiological data obtained in animal studies. And then I tried to model it with 1% of the network size in my local PC. And then I managed to obtain some like interesting dynamics here. But as you have seen that in the previous presentation, results I show you there, if we wanted to use ABR, when we add morphology there, it's always more complex. We have to understand why we're adding it. We're not just tuning the network to mimic what's observed in the experiment. So with this is the starting point. Here I only have a point neural model, a single compartment model for this one. But if we started to add morphology, and it definitely will change again the dynamics. So I think I have this code in the tutorial that you can play with it. Another one is about co-simulation. I've seen that in what we obtained there. Now, if we replace one node with a blue neural network, how it will be like. So I also plotted some sample nodes from TVB, and it really gives a bit different dynamics there. Okay, so that's everything I have for today. So especially for the TCS and the network simulation in upper part, we have it's still under construction. So if you're interested, then you're welcome to join us as a guest student. And for all the work I presented here today, I want to thank my team and my collaborator here in Utrecht and in Paris, as well as the labs I worked before during my PhD and postdoc in Freiburg. So there is where I started to model neuromodulation. So in the end, I think we don't have much time left, but you can keep this link. So that is a tutorial we planned for a student conference last week. It has the Jupyter notebook where you can get started with ABR to learn how to simulate a single neuron or single neuron with complex morphology and the neural network. And I also included the TDCS modeling code there, so you can play with it with even more complex network structure. In the end, I don't know if you got it there, but yeah, so for JSC, we have this guest student program on scientific computing. So if you're interested, you can apply. We have this project listed there. It runs for 10 weeks. 10 weeks, yeah. Okay, thank you. And I'm happy to take questions.
Okay, so it was really interesting and really a lot of things to grasp, like talk about. So I was thinking about when you are simulating the TNS or the TCS simulation, which is the, I mean, before getting to the network level of dollars, how do you model the interaction between, like, for example, like, or if it is done, because maybe it's not done, it's not the focus. Like when you put in the device, in the medical device, okay, 10 milliamperes or 20 milliamperes or whatever the value you are setting up, how does it translate to the model? How do you do that part of physical modeling, if it's done, or it's just supposed to be there?
So there we inject current to the soma to achieve a membrane potential deviation. And the real current intensity, like 10 picoampere or 100 picoampere, cannot directly link to what people used in the experiments. Only the magnitude of membrane potential deviation can be linked there. So there are studies in rats. They propose, okay, how strong the stimulation is, then how like 0.1 or 0.5 millivolt membrane potential deviation is observed. So this value is something we kept in mind when we wanted to model TDCS there. So actually, in another work I didn't show here, we tried to really connect the electric field strength used in vitro experiments to our model. Interesting that we find that in all these in vitro experiments, they systematically underestimated the strengths they use there, because in these experiments, they want to measure it. Usually they use open bath patch clamp sit-up. So this open bath, this relatively large body of electrolyte actually make it impossible to precisely control the electric field. So we published 2 papers there. One, we pointed out, okay, you are doing wrong. So we were rejected for 1 year. Nobody wanted to take our technical review. And the second one is like, it's fine if you don't like it. We design something for you to precisely control the electric field. So I collaborated with bioengineers that happened to be a group. They actually fabricated a chamber. So there we do not have such an open bath anymore. Instead, we have a tiny channel with a rectangular shape at the cross section. And this way we force the current to pass this channel, and then the channel is tiny, so almost the size of the tissue we place there. So we force the current to almost totally enter the tissue. So this way there is no bypass, and then the electric field there is really precise. So with that one, it's really interesting that you cannot directly enter electrode to measure or to do electric field recording there. So we managed to make it possible to do calcium imaging. So we have a well above this chamber, and then so now to observe the effects during stimulation, we just provide another approach to do it, a possibility to do it doing calcium imaging. So I don't know if I can find it here. So yeah, so that is one of the steps that we wanted to stimulation. So if you're interested, so we have this chamber, and it's there. We also have done lots of neuroscience effects, so even different channels to control different intensity which is not modeling work at all, but we calibrated that it's possible to not cause a strong cell death in the tissue, and then actually to be possible to maintain the physiological property of the neurons and then do calcium imaging there. That is one step before we wanted to finally connect the real stimulation parameters into our model.
Can you show the title so I can take a picture and
Okay.
so I have it for to read.
It's called Brain Stimulation on a Chip, a neuromodulation platform for brain slices.
Okay. That's really nice. Because I think it's also like a gap that is like between the real thing and the simulation.
Yes.
In the interface.
Well, I hope after listening for 4 days to inspiring talks, you are motivated to try out some of the things on your own, and you're wondering, how do I get access to HPC? Well, there are several possible ways to do so. In this talk, I will focus on the resources that are provided by EBRAINS partner, which you are eligible to apply to. And one of the major benefits is that all the systems have the EBRAINS software distribution already pre-installed. And that is independent of the type of access that you will get to the systems at Seneca at JSC. There are other ways through EuroHPC and national calls. That's more just for information. The slides you will get at the end. There are some important links that if you want to follow those paths. At Seneca, we have the Galileo 100 system available, and at JSC, we have EUSOF. One difference is that Galileo is reserved for eBrain members and developers, whereas EUSOF is open to anybody. So if you're interested to apply, you should then apply to EUSOF. The application is rather super easy. You have to fill out the template, which you can find in the links that are below here at our EBRAINS wiki, or in the folder case that's for HPC. And you just send it per email to us. This is a rolling call, which means it's open all the time. You can send it in whenever you want, when you're ready. And it is only a technical evaluation. So it's rather fast, and so in a couple of days you will get the access. Before you apply, you should always get familiar with the infrastructure that you're applying to, because that is guiding you then through the process of when you're filling out the application. Because EUSOF will be the one available to you if you're applying through this path, then you should keep in mind that one CPU node has 128 cores, one GPU has one core, and all other systems at JSC have a runtime maximum of 24 hours. So any job that's longer than that is stopped at 24 hours. And super important is the billing model we have per entire node. So even if you start a job and you're using, I don't know, just 8 cores, because that node is now reserved for you and nobody else can use it, we have to bill you the entire node. So then you're wasting the 120 core hours that you have not accounted for. So that's super important to know. And there's a link to documentation if you want to find out more information. What does a good application has? Although this is just a technical evaluation, it's still nice to know what is the scientific question that you're trying to answer. And the most important part, however, is the working plan. How do you want to structure your jobs? So you need to have some kind of a plan. So I want to do X number of jobs for pre-processing, for the main simulation, for post-processing. How much time do you estimate it will take per job? And that is how you then sort of multiply across the row. Across all the columns, and then you sum across the rows, and then you come up with the total number of node hours that you are requesting. It's also important to see that for us, when we're evaluating the application, that the user is aware that they should not be wasting their resources, and they have a plan on how to handle when they're having smaller jobs. So when they are going to occupy only few cores, that they have a plan on how they want to bundle them so that the entire node is being used and not so many core hours are wasted. Sometimes it happens, of course, but it should be, let's say, the exception and not the rule. We do not request that you provide with scalability, but the title of the slide, a good application has, and a good application always has a scalability. But like I said, we're not requesting that, but you should keep that in mind if you're applying to other places. You also need to have a scalability plot so that you know at what number of nodes that you're using your code is the most efficient to. Then other ways to apply for resources are through the EuroHPC resources. This is the joint initiative between the European countries and the partners that are providing the resources. There are several across Europe. You can get more information just by visiting the links that are provided here. They also have different types of open calls. If you're starting out new, the suggestion is always start with the smallest possible, which we the benchmark access. It's up to 3 months allocation time, and it really gives you the time to explore and to get to know the system that you're applying to, and then you can think of applying for development access or regular access, which are then much longer, up to a year. The benchmark access is the deadline for application is the 1st of each month, so you can apply anytime almost. And for regular access and extreme scale are only twice a year, so you can see the dates in this graph. The other way of getting access is through the national calls. The links are provided over here for the sites that are participating in EBRAINS. So again, the ESD would then be installed on those systems. Important to know is that they're open twice a year usually, and the PI that is applying needs to be affiliated, so in the country where you're applying. And one more major difference is usually when you apply through EuroHPC or through national calls, the PI needs to be, the PI of your group, so an established scientist, whereas when you're applying to the resources at EBRAINS that we're providing, then also PhD students that are part, so you directly can apply. It doesn't have to be with your PI, although it's always nice to have the PI, right? Again, always look for the test access at the beginning, because that's the easiest. Usually they are available either every month or they are rolling calls. So you get a small quota of resources to play a little bit, and then when you're applying for the actual call, that goes a long way, that they see that you have tested and you know what you're doing. Yeah, so if you have any questions, please don't hesitate to write to us. Other than that, that was all on my side.